Mia and Cyril for your opening remarks. My name is Sandra Blom and I'm working as senior scientist at the Friedrich Loeffler Institute in Germany. I am member of both the GARA Executive Committee and the IABS Veterinary Biologicals Committee, and it is my great pleasure and honor to act as moderator for today's webinar. Our first speaker is Dr. Douglas Gladue. He is a senior scientist at the USDA Plum Island Animal Disease Center, focusing on rationally designed vaccines for foreign swine diseases, including African swine fever virus. Dr. Gladue is currently a scientific director of the GARA, and we are looking forward to his presentation on the ASF vaccines and current developments at the USDA. Thank you very much, Douglas. The floor is yours.
Sandra, thank you for the introduction. And thank you everyone for joining today's webinar. And today I'm going to talk about some African swine fever vaccines, and I'm going to talk about some of the progress that we made at the USDA as well. I would just like to start off with saying that this is a picture of Plum Island, where all of the vaccine studies at the USDA are done. As you can see, we take a boat to work, and we take a bus, and we end up in our facility in the upper left-hand corner. So with that, I'd just like to give a short overview of African swine fever. This is just an EM picture of African swine fever that was taken at Plum Island. You can see it has double membrane structure. It's a large virus and it has its DNA core. African swine fever is the only member of the ASFV family. It has a large DNA genome of about 170 to 190 kb. There's over 150 genes in all isolates of African swine fever, and very few of these have been studied in detail. ASF can cause a highly lethal, up to 100% of infected animals, to only a subclinical infection. It causes hemorrhage, edema, acidosis, and shock. It can affect wild boar, warthogs, domestic pigs. And the virus host factors responsible for these different outcomes still remain poorly understood. The difference in different strain virulence and the varying host species symptoms and lethality of the same isolate are still widely unknown. We know that ASF can also have a long-term persistent or even a latent infection. And we also know it can have a rapid and efficient transmission among pigs. African swine fever can be passed from domestic pig to domestic pig, wild boar to wild boar, or back and forth between domestic pigs and wild boar. In some areas, there is also a soft tick carrier that remains capable of infecting different animals. ASF has been shown to last several days in feces, months in contaminated pens, and years in frozen carcasses. It has even been shown in pork products that have been salted or dried for over 140 days. And the other method of transmission of African swine fever is to feed swill, garbage, or waste from infected animals. I'd also like to point out that humans can, in movement of illegal imports of meat or meat products, can also cause the ASF to spread. And here's just some over the last couple of years that were in the news. The first one being a seizure at the Canadian border. And this is right after the African swine fever outbreak in China. And there was a whole luggage full of pork products from China. The one in the middle was 1 million pounds of pork that were being smuggled from China through a New Jersey port was seized on March 17, 2019. The next one is just a driver of a truck bought 200 pounds of pork, and they hid them in the engine compartment of the vehicle to try to cross the border. A report that was given last year said that ports of LA and Long Beach, and just these two ports in the U.S., had almost 20,000 pounds of prohibited meat products seized from arriving in China just in the first half of 2020. In these first 5 months of 2020, the inception of prohibited meats in China was 70% over the year before, and this was even with COVID happening. So you can see that different imports of meats are a problem, and this is some examples of what we found. To give a brief history of African swine fever, African swine fever has remained endemic in Africa since its discovery. Outside of Africa, in history, there was only mostly sporadic outbreaks with limited transmission between countries. And these outbreaks that occurred were from undercooked or uncooked pork products that were fed to pigs, and these imported or illegal pork products. And these are the cause of the outbreaks of Portugal, Spain, Italy, and Belgium. There's also been outbreaks that were caused by raw pork waste or garbage at airport or shipping ports, and this was shown to be the cause of the outbreaks in Lisbon, Malta, or Sardinia. However, by the 1990s, Europe was free, and Brazil and Caribbean had also eradicated in the 1970s, and African swine fever remained really just in Africa. This is a picture of what things looked like in 2005. ASF was endemic in southern Africa, Sardinia, and because it was here, and it wasn't really a concern for the rest of the world. However, in 2007, there was a single introduction in Georgia, and this was going to change things quick. The single introduction quickly spread to northern Russia and into western Poland, and remained in this area for a few years. And I want to point out that this area was a relatively low pig-producing area of the world. However, in 2018, ASF was introduced into China. And in the first month, it was spread to 7 different provinces. And just in the first month, there was almost 2,000 infected pigs, affecting 13 farms. And by mid-2019, it was reported that there was a 50% reduction in China's swine herd. And here we are in 2021, and ASF is still a problem in China. So when we look at ASF outbreaks, we can see China in 2018 caused a quick spread of ASF into Mongolia, Vietnam, Cambodia, North Korea, Hong Kong, Laos, into the Philippines, Myanmar and Belgium, Timor-Leste in South Korea, Indonesia. And then in 2020, we had introduction of ASF into India, into Papua New Guinea, into Germany, and in 2021 into Malaysia. So you can see this introduction in China caused a complete change in the picture of ASF. And I think we can say now, in 2021, ASF is a worldwide problem. And it's a worldwide problem that we have no vaccine or no treatment for. There was a report recently that there was naturally occurring variants with lower virulence that were appearing in China. This report gained a lot of attention because it's coming from China, and variants are something that is a concern. But I want to point out that there are 24 genotypes of ASF. All variants currently in Europe and Asia are derivatives of only one genotype. That's genotype 2, and that's from the ASF Georgia strain, and that's from the single introduction from 2007. And I also want to point out that in this report, the variants have very few protein changes, and variants are common with extended outbreaks, and this has been observed in Eastern Europe with ASF Georgia. For example, in Latviia, Estonia, in these 2 reports. I also want to point out that a lot of the variants that are identified in this manuscript have already been identified in other parts of Europe. So they're not necessarily new variants, but they're similar variants that we're seeing in other areas with this outbreak strain. The next thing I want to point out is how ASF had effects on the global pig supply. You can see here in this graph that China produces about 1/2 of the world's pork supply, with the European Union coming in second at about 25%. So what happened to the swine inventory after ASF introduction? Well, there are a few reports where Bulgaria, for example, so there was a 25% swine inventory decrease. China reported 50%, Philippines 25%, In Vietnam, the estimates were 35 to 45%. So this large decrease in swine inventory shows us that ASF is a threat to the stability of global swine industry and to the world's available protein supply. Currently, it's been reported that there's a second wave of ASF that's occurring both in China and in other parts of Southeast Asia. So what were some of the ASF vaccine gaps? I'm just going to highlight a few of them that we talked about at our dairy meeting in Italy. Protective immunity from viral proteins are unknown, and not knowing this makes it very hard to make subunit vaccines, where any attempts have failed or gave only a modest protection rate, and successful subunit vaccines have been to low virulent strains. And these require large doses, and these large doses are in general commercially unviable. Inactivation of ASF does not offer protection even at high doses. This means that an inactivated vaccine is just not possible. And this has been known for quite a few number of years. There's no live attenuated vaccine commercially available. The mechanisms of protection involved in experimental vaccines are unknown. And the cross-protection studies between different strains of ASF have been very limited. Live attenuated vaccines are aligned with the development of recombinant ASF, and this is a very time-consuming process. So when we talk about live attenuated vaccines that have been shown to protect against homologous challenge, how do we produce these attenuated isolates? Well, there are reports with low virulence field isolates, reports with viruses attenuated by tissue culture passages, and viruses with genetically engineered deletions. So I'm just going to briefly talk about these three options here. It was first shown in the 1980s that asymptomatic carriers were resistant to homologous challenge, bringing the idea that a low virulence field isolate could be used as a vaccine. And there's several reports since then that looked at different low virulence field isolates, and they tested them against homologous challenge. And in all of these cases, the same thing happened. All low virulent field isolates had some level of residual virulence. That means that vaccines based on low virulent field isolates do need some sort of further attenuation or some sort of vaccine safety, because this residual virulence is not something that we want to put out into commercialization. So the next one I want to talk about is viruses attenuated by tissue culture passages. And there have been several attempts at this. I'm going to talk about the study done at Plum Island, where we took the Georgia isolate and we adapted it to Vero cells. And what we found is as we passed Georgia isolate and Vero cells, by the time we got to passage 60, we had 5 out of 5 animals survive a low dose, 10 to the 2. And by the time we got to passage 110, we had 5 out of 5 animals survive a dose of 10 to the 4. So we were able to attenuate ASF by cell culture passage. However, when we looked at the genome of ASF, we found that we had 2 very large deletions on either end of ASF Georgia. And this virus adaptation to growing cell lines caused these dramatic genomic changes that resulted in the inability of this attenuated virus to replicate in swine, therefore making this cell culture adopted to Vero virus not a vaccine candidate. So next, I'm going to talk about viruses with genetically engineered deletions. I'd first like to just show there was a very good review by the Dixon group in June 2019. And in this review, they show that so far in the history of ASF, there's 16 different gene deletions. 7 genes are associated with virulence, and 5 of these genes attenuate field isolates. So I'm going to talk about live attenuated vaccines that protect against the current Georgia strain and viruses with genetically engineered deletions. The first one I'd like to talk about is ASFG with a deletion of the gene 9GL. So 9GL was first identified as a virulent determinant by Lewis et al. In the Malawi strain. It was protected, and when we deleted it from Georgia, we found that it was protected at a single low dose. However, it was virulent at higher doses, which was something that was not seen in the initial deletion of 9GL by Lewis et al. So we knew the Delta 9GL vaccine platform would need further attenuation. So we took a gene UK, which was first identified as a virulent determined by ASFV, and we deleted this in our Delta 9GL platform. And what we found was deletion of UK increases virus attenuation at least 100x. It increases protection when administered once at 10 to the 4. And at the time, this was the only double deletion live attenuate vaccine that protects against current outbreaks here in Georgia. The next gene I want to talk about is CD2. And Fernando Rodriguez's vaccine, which is deletion of CD2 and BA71. This is a genetically different virus than the Georgia virus, however, has a very unique ability that it's able to grow well in host cells, making it an attractive vaccine candidate. It was attenuated in swine at doses 10 to the 3 to 10 to the 6. There was fever present in some animals. Interestingly, challenge animals were able to survive both BA71 or Georgia challenge. Now, we looked at this CD2 gene in African Swine Fever, Georgia, and when we deleted CD2 from African Swine Fever, Georgia, we found that there was no effect on virulence. It was not attenuated even at low doses, and this was also repeated at Harbin in the study by Chen et al. And both of us found that deletion of CD2 in the Georgia strain of ASF does not reduce virulence. The next family of genes I'd like to talk about is the Multigene Family. And a lot of times these are referred to as Delta MGF. However, deletions in this area can be very different. I want to point out there are a lot of Multigene Family genes in different isolates of ASF. But there's 5 family members, 100, 110, 300, 360, and 505. And here's a nice tutorial from the Viral Bioinformatics Research Center, which shows all of the MGF genes and all the sequenced ASF genomes. And you can see here that there's a varying number and combination of MGF genes in these genomes. And there have been several reports where there have been different deletions and different virus determinants of virulence within this area. So this is an area of the genome of ASF that has been historically studied in several different genomes. However, I'd like to point out one specific deletion, and I'm going to zoom in on the small area of this region of MGF genes. And this is the ASF Delta MGF deletion that was done by the USDA. It's a deletion of 6 MGF genes. It's protective at a single low dose. So there's no clinical symptoms at higher doses, only about 1 day of mild fever. To this vaccine candidate, we deleted another gene, Delta of 9GL. And when we took the 9GL deletion and we added it to our MGF gene, we did still maintain full attenuation. However, we had no replication in swine. And this shows that the addition of just a single deletion to this MGF deletion was not a vaccine. Another deletion in this area was reported here by Chen et al. And this is taking the MGF deletion from the USDA vaccine platform and adding a deletion of CD2. In this report, it also confirmed our report that CD2 by itself did not attenuate the virus. However, the 7-gene deleted ASF is protective. In this report, they tested reversion to virulence by 5 passages in pigs. They also tested this platform in pregnant sows, and currently this is being tested in field trials in China. There are other MGF deletions, and these are 2 recent reports from earlier this year. There's a deletion of MGF 110-9L, and this was attenuated in swine at very low doses of 10. Challenged animals did survive. The vaccine was detected in nasal swab, showing possible shedding, and there was no report of any higher doses being tested. Another MGF deletion was reported, which is deletion of 5057R, and this was also shown to be attenuated in swine at, again, very low doses of 10, and higher doses or vaccine efficacy was not tested in this report. I also want to point out that we've done some other deletions in this area at the USDA. We deleted MGF genes 13 and 14 within our MGF vaccine deletion. We found this deletion to be virulent. So a smaller deletion of only 2 genes was fully virulent. We also deleted gene X69R, and this was also virulent and had no effect on the virus. And I also want to point out that in our Vero adaptation, that this deletion, along with a deletion called L7L to L11L, encompassed part of our MGF vaccine deletion, but not all of it, and some other MGF genes. And this was attenuated and was not a vaccine. There is also from Pirbright a delta MGF deletion that's larger than the MGF deletion done at the USDA, and this was done in the genome of Benin. And in this report here, it was shown that it gave partial protection. So I point this out because a lot of times we're grouping delta MGF deletions as the same, but I think it's pretty obvious here that we have some specific deletions that are vaccines. When you add genes to it, and additional deletions, the vaccine efficacy may change. So not all delta MGF deletions are the same, and only specific MGF deletions are vaccines. Additional deletions or genomic changes can severely affect vaccine efficacy. And I also want to point out that these additional deletions or genomic changes will go undetected if the deletion strains are only analyzed by PCR, which is why it's very important to fully sequence any experimental vaccine. Another vaccine that was reported is deletion of the L7L to the L11L. And this is deletion of the same genes in the right side of the genome that were found from Vero adaptation in both African Swine Fever, Georgia, and BA71 adapted ASF. It was attenuated in swine at 10 to 3 to 10 to 6. The challenged animals survived, and however, fever was present and death of one vaccinated animal did occur. So what that tells us is that this deletion of L7L to L11L, further attenuation is needed to consider this a vaccine candidate. So it became pretty clear that looking at all the historical determinants of virulence and deletions that we need to find new genetic targets in African Swine Fever, Georgia. So here we created a custom computational pipeline that takes into account different sources of bioinformatics and functional genomic data. We identified 3 new ASF protein targets: EP152R, which is a CCL2-like protein that could not be deleted, L83L, which is an interleukin-1 binding protein, and that didn't seem to have an effect on virus virulence, and I177L, which is predicted to be an immune modulatory protein. And our I177L gene deletion was published last year, but I'm going to go over some of the results now. So we were able to successfully delete I177L, and as you can see here, we had a slight growth reduction in macrophages. However, we were easily able to get high titer stocks from this deleted strain. We showed that I177L is a late transcribed gene. We showed that I177L deleted from ASF is completely attenuated in swine. And this vaccine candidate showed no clinical disease, including fever, even at high doses of 10 to the 6. We found that animals that were given delta I177L did have a viremia that was present, and this viremia correlates with the different doses. We also shown that we had an antibody response that correlated with this viremia in animals with different doses of delta I177L. We wanted to conduct a virus shedding experiment and to determine if this virus could be given transmitted with vaccinated pigs. So sentinel pig was housed with vaccinated pigs. We found no detection of vaccine virus in the sentinel pig, as shown here in red, the bottom line red in all 3 groups. That means that there's no viral shedding of ASF delta I177L, and the vaccine virus does not spread to unvaccinated animals. And this increases the safety profile of our vaccine. When we challenged animals that were giving delta I177L with African Swine Fever, Georgia, we found that Delta I-177L protects swine against challenge. Even at low doses of 10 to the 2, we were easily able to achieve 100% protection. I also want to point out, we looked at differential PCR of I-177L, and after challenge, we found that there was absence of challenge virus. We found absence of challenge virus in 9 out of 10 animals that received 10 to the 2 vaccine, and 5 out of 5 animals at doses 10 to the 4 or higher. And this tells us that African Swine Fever, Georgia, Delta I-177L has 100% sterile immunity at doses of 10 to the 4 or higher. We next wanted to see if we could deliver our vaccine by the oral nasal route. And these results were recently published about last week. As you can see here with the oral nasal on top, the intramuscular on the bottom, and both oral nasal and intramuscular vaccinated pigs remain clinically normal with no residual temperature. Interestingly, when we looked at viremia in both of these groups, we found a very low level of viremia in oral nasal vaccinated pigs. We found a higher viremia of up to 10 to the 4 to 10 to the 7 in pigs given an intramuscular injection. This viremia persisted in some animals even to the day of challenge. Next, we looked at if oral nasal inoculated Delta I-177L animals could be protected from African Swine Fever, Georgia. And as you see here, we had 5 out of 5 animals survive, so we still maintained 100% protection when this vaccine candidate was given oral nasally, similar to what we observed when it was given intramuscular. When we looked at the antibody response, we found that the oral nasal vaccinated pigs did have an IgM response that occurred slightly earlier and longer. However, both oral nasal and intramuscular pigs had very similar IgG responses. So therefore, we see no major differences in antibody response, no matter if it was given oral nasal or intramuscular. That makes oral nasal delivery of ASFG Delta I-177L a viable delivery method. So just to summarize with Delta I-177L, it induces protection even at an intramuscular dose of 10 to the 2. It lacks residual virulence even at an oral nasal or intramuscular dose of 10 to the 6. It does not induce virus shedding in either group. It induces sterile immunity, has the highest safety profile for any live attenuated vaccine that protects against African Swine Fever, Georgia, and to date is the most promising experimental vaccine. However, it has its limitations. Similar to all recombinant experimental vaccines made from field isolates, it only grows primary swine macrophages. Primary cells have commercial limitations. They require donor animals, time-consuming to isolate, regulatory concerns, expensive, and inability to make a master cell line vaccine production. And this was considered one of the major gaps that we identified at the last in-person donor meeting, and that was that ASF lacks a cell line both for vaccine production and diagnostics. Primary swine macrophages were the only cell line to support ASF. Again, requires donor swine, it's very time-consuming and costly, and also it cannot be rapidly scaled up in case of an outbreak. So last year, we solved one of these problems where we identified a commercially stable cell line for the identification of infectious African swine fever in clinical samples. And here we show MA104 cells. And as you can see, in MA104 cells or macrophages, you're able to see rosette formation. We were able to do antibody staining, which was a plus for MA104 as they had almost no background. So it makes it very easy to antibody stain for ASF. And we found that when we tested different genotypes, the MA104 detects different genotypes of ASF. It's about 1 log less sensitive than macrophages, but MA104 cells are readily available through ATCC and grow under typical cell culture conditions. It's also important that they can divide rapidly and be stockpiled and frozen. So MA104 cells are a suitable cell line for detection of live African swine fever. And here's just a link for a detailed protocol in case anyone's interested. So to try to solve the vaccine cell line problem, we did a systematic screening of commercial cell lines. And we were looking for a cell line that could support vaccine production similar to macrophages, with at least 5 logs growth, a stable ASF genome, and similar efficacy as non-adopted vaccine. So the cell lines we tested at Piatic, what we found was that we took a fluorescent reporter virus to monitor infection, and almost all cell lines are somewhat infectable at a very low level that we tested. However, ASF is lost with 3 to 5 line passages in most cells. Adaptation typically resulted in genome instability and large or multiple deletions in the genome, and increasing instability with increased passage in various cell lines. However, I'm happy to announce that our paper was accepted a few days ago, and we were successful in screening of cell lines, and we found an in-house cell line pipette cells, Plum Island porcine epithelial cells. At 6 passages, we saw a clear cytopathic effect. By the 7th passage, we expanded to a stop. And successive passages increased virus yield. As you see here, by pass 7, we had some growth of our vaccine candidate, but by pass 11, we had growth that was similar to that in swine macrophages. At passes 11, we achieved a 10 to the 6 to 10 to the 7 titer, which is a 5 to 6 log increase in titer. It's important that pipette cells do not require any special media requirements, are rapid cell growth, and have characteristics for potential vaccine production. When we looked into genome stability, we found that we sequenced the virus at passage 7, 11, 20, and 30, and we found there was a deletion in the left variable region of the genome. We call this deletion delta LVR. Modification of 10 genes occurred where we deleted 8 genes and fused 2 genes. We had 7 MGF genes deleted through 66L, 8L, 9L, and 10L, MGF 300, 1L, 2R, and 4L, and gene X69R. We also had the fusion of 2 partial ORFs, and that's of genes 364L and 11L. It's important to say that these genomic changes appear to be stable up to 30 passes in pipette cells. However, we did observe 1 point mutation in E119L that occurred between 20 and 30 passages. However, this point mutation was pretty conserved serine to 30. So here I just want to show that the deletion of MGF genes in our delta I177L, delta LVR are different genes than the MGF deletions in vaccine candidates or other cell culture adopted viruses. The pipac adaptation mutation is different. It's to the left, and you can see here all of the other MGF deletions are to the right of this deletion. When we compared this area to other isolates of ASF, we found that all isolates of ASF over sequence have all 3 of these MGF 300 genes, and most isolates had the other deleted MGF genes, with all 6L and 8L being present in all isolates. So this was interesting, that these genes, for some reason, appear to be highly conserved and required in all isolates of ASF. However, they were able to be deleted in our adaptation studies. So the next thing, and the question is, what is the efficacy in swine? When we compared our delta LVR and our parental, I177L, we found that they had similar low levels of viremia at doses 10 to the 2 to 10 to the 6. When we co-housed a sentinel animal, it remained negative, and did not, the adopted virus did not get past the sentinel animal. When we looked at the antibody response, we saw that occurred as early as 7 days with our I177L LVR, and this was similar to the parental I177L. When we looked at that vaccine efficacy, we found that our adopted virus induces 100% protection at 10 to the 2, which was the same as the parental virus, so it maintained vaccine efficacy even with this deletion and adaptation of pipette cells. Just to conclude, our adopted virus, or delta I177L, delta LVR, induces protection even at low dose of 10 to the 2, lacks residual virulence even at doses of 10 to the 6, does not induce virus shedding, can induce sterile immunity, and maintains the safety profile of our delta I177L vaccine. It grows to high titer in pipette cells, and pipette cells are a suitable cell line for ASF vaccine production. And this is the most promising experimental vaccine that protects against African Swine Fever, Georgia. I'd also like to point out that our adopted virus and our other vaccine candidates, Delta I-177L, Delta MGF, and Delta 9GL-UK, have received select agent exclusion. And this means that the vaccine will not require oversight from the select agent office and that it can be produced in BSL-2 in the United States. So where are we now in our vaccine status? Our Delta I-177L and our Delta LVR have the highest efficacy and safety profile of any live attenuated vaccine for African Swine Fever, Georgia. The USDA has licensed 4 ASF vaccine platforms like Delta 9GL-UK and our MGF to 5 licensees. With Zoetis and 4 undisclosed partners. Our Delta I-177L has been licensed to 3 licenses with 6 under negotiation. And that's Naveco in Vietnam and 3 undisclosed partners. And our Delta I-177L, Delta LVR has been granted 2 licenses, and there are still 7 under negotiation. What commercial partners and how are our research agreements going? With our 9GL-UK, we have an undisclosed partner, and it's up to them to report the results. For our Delta I-177L, we partnered with Naveco in Vietnam, and Naveco has repeated our vaccine studies with very promising results. It's performing required safety studies for licensing, and our Delta I-177L has been shown to be effective in their hands against the Vietnamese isolate and has been tested to be effective in both local pigs and commercial breeds. So what are the current vaccine gaps? Standardization of methodologies to test efficacy of vaccine candidates. And this is challenge methods and doses are very different between different laboratories and different studies, making it very hard to compare different studies. Some of the recent vaccines are not fully sequenced and relied only on PCR for purity. All of the USDA vaccines are fully sequenced and don't have any off mutations. It's unknown the onset of duration of immunity. Attenuation stability and long-term vaccine studies need to be conducted. Minimal protective doses by different routes of inoculation are not known. Currently, there is no DEVA vaccine candidates. And understanding the different outcomes in virus virulence between different isolates is necessary, and understanding how the same gene deletions can have differential effects on reduction of virulence is important. And also for defining regulations for testing safety of ASF vaccine candidates. And finally, I'd just like to show a picture from our visit to Naveco. And this is a visit with myself, Cyril Day, and Manuel Borca. And here's a picture of us with the Naveco team. This happened just before COVID hit when we started our collaboration with them. I also think it's important to acknowledge the group at Plum Island that has been working on our African Swine Fever vaccines. Manuel Borca, we have 2 new cancers, Sal Medina and Eddie Young have joined our group. Elizabeth Ramirez and Elizabeth Bruno do a lot of our recombination studies. Yushi Rai did the adaptation of our vaccine candidate and discovered MA104 cells for diagnostics. And also want to point out, Lauro did some of the real-time PCR, and Nayeli and Sarah are responsible for our production of weekly macrophages and for keeping up with all the other requirements in the lab. I also want to point out that there were a lot of people that are currently not with us at Plum Island that have left for other research jobs, and they all contributed, at least in part, to where we are today with African swine fever vaccines. And finally, I'd like to acknowledge that some of these projects were partially funded by the National Pork Board and the Department of Homeland Security. And I'd be happy to take any questions, or I'd be happy to take any questions by email as well, with my email address right there. And thank you, everyone.
Thank you, Doug, for this overview and providing us with promising results.